Okay, so for this particular conversation, you guys will want to have your cells packet, and we are continuing to build each week and work our way up to talking about organs and body systems and the human as a whole. So this week's conversation is going to focus on the cell and the inner workings of what has to take place within the cell for it to provide the body with a lot of different material. So, the one thing that I think people kind of have in the back of their mind, or a couple of misconceptions that people have about the cell, is that number one, it's just a building block, and we put a bunch of them together and we build an organ, or we stack them all together and we build bones. And there's so much more going on in the cell than just that. Likewise, a lot of people have this misconception that the cell is just kind of this open space inside and things are freely floating and moving around, kind of like a bathtub of water and things just float and move where they want to. Not the case whatsoever. So, when we look at the cell is actually a tiny factory. And yes, it is a building block, but that's not the only thing that it does. Within inside each cell, you have products that are being produced. And once those products are produced inside the cell and they are ready to go, then you have this very in depth process of taking that product and shipping it to where it needs to be used. Sometimes that's going to stay in the cell. So the cell makes the product and it needs to be used inside the cell. Sometimes it has to be delivered somewhere else in the body. And so once it's produced, it's pushed out of the cell completely and it is going to then travel to where it needs to be used in the body. So it just depends on the product and what the body needs at the time as to the outcome of those products that are made within the cell. But if you've kind of had that thought in your head that it's just you put a bunch of them together to build something, yes, that's right to a certain extent, but also start thinking of the cells as factories and they are constantly producing materials. And that can change from day to day, depending on what you need within your body. That will vary. You might produce a certain material one day and not need as much the next day, and so the cell will start producing something else. So it has that ability to read what's going on in the body, what the needs of the body are, and adapt to that. So when we look at the cell, we have the two cells that are going to give rise to everything else, which are the egg cell in the female body and the sperm cell in the male body. Now, the unique thing about these two cells is that you have the smallest cell and you have the largest cell. So, if you were to take every single type of cell in the human body, no matter if it's male or female, and you were to rank them, sperm cell is going to be the absolute smallest, and then your egg cell is going to be the absolute largest. The egg cell from the female is the only cell that is actually large enough to be seen with the naked eye. So that is a unique quality or trait that we have with the egg cell. But it takes the union of a sperm cell and an egg cell to come together to start the process of developing a human, and then that process of those cells increasing in number so that you can get all the parts that you need for a fully functioning organism. So when we look at that process of the sperm and the egg uniting, once those two come together, you have two cells. They will start the cell division process. Two become four, four become eight, eight becomes 16. And you continue on through that process. And in those early phases of cell division, when we are doubling in number, those cells don't have a purpose yet. They are what we would call primarily stem cells. And at that point, it's just increasing the cell number. And at some point during the process of development, all of a sudden, those cells will start to transition. And you will get a group of cells that are going to ultimately become your heart. And they will start getting all of the special features they need in order to become heart cells. You'll get another cluster of cells that is going to form your stomach, and they will start getting all of the special features that they need in order to be your stomach. So early on, stem cells, that just refers to those cells that are kind of blank slates. They could become absolutely anything that the body needs. And as we continue on in development and we get those cell numbers built up enough, that's when you're going to see the process of specialization, where those cell groups are going to get their orders, so to speak, as to what part of the body they're going to become. Now, as much as we know about the human body, this part is actually still a huge mystery. They don't know exactly what causes the cells to turn or convert into a particular body part. And that's really what they're trying to harness. If they could figure that out within the body, how you have these cells that are blank slates, and all of a sudden, where does that information come from? What triggers them or turns them on to start developing and become a certain organ or a certain part of the body? And scientists very heavily believe that once they're able to understand that and how that actually works or takes place within the cells, that a lot of the problems we have, a lot of the diseases and disorders that we face as humans right now, we would be able to take care of, because we could then take stem cells and we could program them to be the type of cells that we want them to be. So organ donation would not be as big of a problem, because we could harvest these stem cells and we could then program them to be exactly what we need them to be. The controversy with stem cells is that the best place to get them is from that early point of development, when you have a large cluster of cells that have not been given their information yet. And that is going to be in a developing embryo. As adults, we have stem cells all over the place. The problem with our stem cells is that they are embedded and ingrained within our existing tissues and our existing body parts. So we do have them within our own bodies, but because of how they are scattered throughout, we don't have large numbers of them grouped together, and it would be much harder to get to them, and it would be much harder to get them in the numbers that we would need them in. So stem cell research, very controversial because of that particular fact. If you look at the second slide, it talks about the cell membrane. The cell membrane has a very specific design in order for it to do its job. The cell membrane has the role of separating the cell from the rest of the body. So you have this layer around the cell contents that are designed to maintain everything inside. And at the same time, the cell membrane is going to allow for material to move in and out. And that goes back to those products that the cell is producing. Once those products are made, some of them are going to get shipped out of the cell. We need to make sure that there is an easy way to do that. And so the cell membrane has to be designed in a very special way to, number one, hold everything together, hold all those contents inside, but also allow materials to leave when they're ready. So the design, the major part of your cell membrane is going to be made out of lipids. And a lipid within the cell membrane has a round head and two tails coming off of it. The head portion, the round head, loves to be exposed to water, which is great when you're in the human body, because if you think about most of our internal contents, they are going to be in a liquid environment. So the tails, on the other hand, hate water. They do not want to be exposed to it. So this really helps with the design of the cell membrane, because it's two layers of these lipids, and the heads always face the outside, the tails always face each other. So you get this layer of lipids that faces the outside of the cell. You have another layer of lipids that faces the inside of the cell, both areas usually very high in liquid content. So that's the way the heads turn. They want to be exposed to that liquid. Tails, on the other hand, like I said, they hate water. They're hydrophobic. Hydro means water, phobic means to have a fear of. So the tails will always stay turned to the inside, and you get this double layer of lipids holding all the contents in the middle and then creating a barrier from all the contents or material surrounding the outside of the cell. Now, the thing about the lipids is, because you have the hydrophilic, the heads that love to be exposed to water. Hydro means water, phil means to love. So they will always position or turn themselves to face those liquid areas, but they're not secured together. And that's very, very important for the design of the cell. If we're moving material in and out of the cell all the time, we can't have those lipids being fused to each other. So just the fact that hydrophilic always will move to the water, hydrophobic will always move away, we can maintain that cell membrane without having to have the lipids actually fused or bonded together. The lipids will allow for materials to pass in between if they're a certain size. If they're small enough, we can just slip material in between two of the lipids and it won't cause any problems. If you have materials that start to get a little bit larger that need to be moved out of the cell, then we have to find another way to get them out. If you move to slide 3, it talks a little bit about how we get those larger things out of the cell. So main component of a cell membrane, always the lipids. But you do have two other very important parts of the cell membrane, and one is going to be a protein, and the other one is going to be a carbohydrate. Now, the proteins are actually going to create doorways. So if you guys think about a room, if you guys are going to move into a room, you have to have an entryway, a way to pass in and out of that room. And it has to be large enough for you to move in and out. That's what these protein channels will do within the cell membrane. For those items that are too large to fit between the lipids themselves without ripping open the side of the cell, we are going to embed these protein channels, and they work like a doorway. Very much like a door. If you close the door, you can prevent things from moving in and out, or you can open the door, which will give materials free range to move in and out. And those protein channels work exactly the same way. They can be opened, let things pass across, or they can be closed and prevent things from moving if the cell doesn't want that movement to happen. The carbohydrates, on the other hand, are going to be mainly on the surface of the cell. And the carbohydrates work very much like flags. That is the best example that I can give you when you think of those carbohydrates. They are on the surface and they are going to do a couple different things. Number one, they are going to let your body know that that's something that you made. So it is a way for your cells to recognize each other. If you think about the environment that these cells exist in, it's dark. They have no way to see each other or visualize each other than these carbohydrate flags. And when two cells are bumping around in this dark environment, when those carbohydrate flags touch each other or touch another cell, you're immediately able to recognize that that is something that your body produced. You think about going to the moon. What did we do when we went to the moon? We stuck an American flag on the surface of the moon. So we could recognize that we were the ones that had been there. And likewise, your cells use the same sort of system. They put these carbohydrate flags on the surface of the cell so that as cells are bumping into each other, they recognize one another. If you get a foreign invader in your system, whether it is a harmful foreign invader or it's just something foreign that got into your body that's not going to cause any problems, they will have their own carbohydrate flags, but just like a flag from a different country is going to look different than ours, their carbohydrate flags are built different, designed different. And so now when your cell comes in contact with their carbohydrate flag, it's not going to recognize it. And we immediately know this is not something we produced. We need to find out why it's here, what it's going to do to our system, and then determine how we're going to get rid of it. The other thing that the carbohydrate flags will do is during periods of growth and development, those carbohydrate flags tell your internal parts how much space they have to grow. So when you guys, if you think about your internal organs, they overlap, they touch each other, they come in contact with each other. There is no gap or space between your heart and your lungs or your stomach and your liver. They do touch. When you have a growth spurt or a time where you grow and develop, as you expand upward, your organs have more room. They also need to increase in size to accommodate the needs of your body. And so they will start producing cells to increase in size as well. And those carbohydrate flags on the surface of your stomach and the surface of your liver, when they start touching each other, they know that they have increased in size to the right amount and that they need to stop producing more cells for that time period because there's not enough room anymore. You have another growth spurt, and those carbohydrate flags maybe aren't as touching as much, not coming in as much contact. They know they need to start increasing in number again. They will increase the number of cells, And as they grow bigger, those carbohydrate flags touch again, and now they know that they've taken up their space. And so that's just a way that your body can monitor how much your organs should be growing, how much they should be increasing in size, and when they've taken up their space. If you move on to slide number 4, it gets into the organelles. Organelles, very true to their name, they're tiny organs. And if you think about everything that your body has to do in order to keep you alive, your cells have to do the same thing. So there are a lot of counterparts in our cells that are similar to the organs we have in our body. So we have to have a brain inside of our cell to monitor everything that is going on. We have to have a way to get rid of waste product. We have to have a way to create that ATP and that fuel source to keep our bodies going and our cells going. And so as we go through the organelles of the cell, like I said, you'll see a lot of overlap in what your organs have to do for your overall body. So if we start at the top, you have cytoplasm. Cytoplasm is a gel-like material, and it is going to do a couple different things. It is going to create a semi-liquid environment inside of the cell so that as those cell products that have been made are getting shipped out of the cell, it's a little bit easier to move them with that liquid environment. And you also have other materials that are going to have to be delivered from point A to point B within the cell, and that liquid environment helps with that. The other thing that cytoplasm is going to do is it is going to help maintain the shape of the cell. We think of cells as being that nice round circle, and for the most part that is true. And so the cytoplasm just helps to hold that shape that we think of. The next one that you have on the list is chromatin. So one of the most important things that is housed within most of our cells, not all, but most of our cells, is going to be our DNA. DNA is the blueprint for how you are made. Has all of the instructions from your eye color to your hair color to how tall you're going to be. Everything that makes you you is held within that DNA. So as the cell is growing and developing, your DNA will take on different forms, and chromatin is one of those forms. So chromatin is the stage when your DNA is very relaxed. You're not in the process of cell division, and so the DNA gets very spread out. Does not have that typical X shape that we like to draw when we talk about chromosomes. It just usually is depicted as having like a speckled appearance because it is so spread out that it really doesn't have any shape or form to it at that point. So chromatin is just one of the forms your DNA can take on, and that is the form that indicates your cell is not in the process of cell division. The next one that we have are the ribosomes. Ribosomes are tiny little round structures that you have in 2 locations of the cell. Some of them are going to be free floating in the cytoplasm, and others of the ribosomes are going to be attached to the endoplasmic reticulum, which we're going to talk about here in just a minute. So ribosomes are these tiny little structures that are made of protein. And protein is found in pretty much every part of our body. If you think of any cell, your cell membranes have those protein channels, your DNA has protein in it, your muscle is protein, your tissues, your organs, there's going to be protein involved in making all of those. And so the protein that you guys put into your body through your diet, a lot of that does not go to being used as an energy source. It is going to building material within your cells. So these ribosomes help with that protein production within your cells. And so that's why we have it in 2 locations, because we want to make sure that that protein is readily available to be used. The next one on the list is the nucleolus. Nucleolus, best example, so it's contained within the nucleus. Nucleus is housed in the middle of the cell, and that is for protection. The nucleus is the equivalent of the brain. Very, very important. So we always hold it, I shouldn't say always, almost always, we hold it at the center of the cell so it can be best protected. The nucleolus is inside the nucleus. And if you are kind of a visual learner, think of an egg. You crack open an egg, you have the clear white part, that's the nucleus, and then you have that smaller dense region in the middle, the yolk, that would be the nucleolus. Nucleolus has a couple of different jobs. It's another place where we can make protein. And as you start to see protein show up in multiple places, we're making it in the ribosomes. Now we're seeing it being made in the nucleolus. That's telling you that high importance that we have that protein production happening. And then it's also going to be a site of RNA. RNA is a component of DNA. It works with DNA to copy information and get it delivered to the parts of your body. And that will be a conversation that we have a little bit later on. The next one is the endoplasmic reticulum. A lot of times, textbooks will abbreviate the endoplasmic reticulum down to the ER. If that makes sense to you, feel free to use the abbreviation ER. That is perfectly fine. If you've never heard of it or dealt with it before, maybe you want to use the entire name until you get a little bit more familiar with it. The endoplasmic reticulum is a structure. It's defined as being a network of membranes, which is very similar to the definition that you usually see with the Golgi apparatus. What sets the two apart, number one, the endoplasmic reticulum comes in direct contact with the nucleus. So that's how you can distinguish between the two right off the bat. If it touches the nucleus, then it's the ER. If it does not come in contact with the nucleus, then it's a Golgi apparatus. A lot of times when you look at them on pictures, they look very similar because they are made of stacked or layered membranes. So using that point of reference as to whether or not it comes in contact with the nucleus can be helpful. With the endoplasmic reticulum, it fits right up against the nucleus, and that's another place within the cell where you have a lot of material being made. So having that endoplasmic reticulum right up against the nucleus, as products are made, we can put them right into the ER, and we can channel them using those membranes and direct it where it needs to go. Sometimes it's going to be delivered to another part of the cell. Other times it is going to be pushed out of the cell. Just depends on where it needs to go. Best example I can give you is to think of the endoplasmic reticulum as hallways in a building. When you guys came into the building, you had to go through the doors, you had to go down the hallway, and that hallway directed you to the classroom where you needed to go. If at any point during class you need to go to the restroom, you are going to get up, you are going to go out the door into the hallway system, and you are going to use the hallways to navigate your way to the bathroom. The endoplasmic reticulum does the exact same thing. It is a series of hallways within the cell that is going to direct material and get it to where it needs to be. Nuclear envelope surrounds the nucleus, somewhat similar to the job of the cell membrane, but with the nuclear envelope, it has thousands of pores across the surface so that as those products are being made, they can simply move out of those pores, or material can move in through the pores, and we can get that exchange in and out through those tiny little holes or openings that you have in the nuclear envelope. Now, despite having those nuclear pores, the DNA, which is housed inside the nucleus, 99% of the time, DNA is always going to be in the nucleus. Those pores are too small for the DNA to be able to move across them. And so even though they're open all the time, because of that size difference between the DNA and the pores, we don't ever have to worry about that DNA being able to leave or slip through those holes. The last one on that list is the vacuoles. Vacuoles are round structures that you have within the cell, and they are going to hold water. So they are kind of little storage tanks. They hold water within them. And I could say that and I could leave it at that, and people would memorize it and know that the vacuole holds water. But there's a reason to it. There is a very important reason why we have to have these storage tanks within the cell, and we can't just let that water go wherever it wants to within the inside of the cell. The reason for that is the cell has to be compartmentalized. It is not a free open space. There are some things, yes, that can move through the cell. They are not physically attached to anything, but majority of the components in your cell are going to be in a fixed location. And the reason for that is because we have products being made. A lot of those products have to be made through chemical reactions. Those positive and negatives that we talked about in our Biochem week, opposites attract. And sometimes your cells need a very specific chemical reaction to happen. But if you had all the positive and negatives just floating around, any of the positives, any of the negatives would be able to interact with each other. The cell is set up very specifically, compartmentalized, has very specific locations, and we keep the positive and negatives that we need to interact with each other in certain locations so we know exactly what chemical reaction is going to be carried out. Again, if you are kind of a visual type learner, think of your house. Your house is compartmentalized. You have a room in your house where you cook your meals, your kitchen. You have a room in your house where you watch TV in the living room. You have a room where you sleep, the bedroom. Very compartmentalized. Each room has a very specific function that it carries out within your house, and the cell is no different. We need to make sure that those chemical reactions happen when and how, that the wrong positive and negative materials are not allowed to interact. And by having those specific spaces built within the cell, we can do that. Now, where do vacuoles play into that? Because these chemical reactions need to take place, we can't dilute them. And if water was allowed to flow freely throughout the cell and we didn't have a way to contain it, we would dilute the environment within the cell, and some of those chemical reactions wouldn't happen the way that they need to. You think about if you've ever made Kool-Aid. If you follow Kool-Aid according to the directions, it turns out just right. But if you were to take that Kool-Aid and then add another liter of water to it, and you taste it, it's not going to taste right. It's not going to come out the way it's supposed to. Likewise, if we leave all that extra liquid in the cell, those chemical reactions won't happen the way they're supposed to. Next set of organelles, we have the vesicles. Vesicles are another structure that are going to hold liquids. And these are typically liquids, but they're not just water. So sometimes it's a liquid waste product, things like that, that you have built up in the cell. So vesicles are a little bit more specific. It's still liquid, but it's those liquids that are not just water. Microfilaments are these little protein rods, and they work very similar to a skeleton. So your cells don't have a support structure like our skeleton, where bones are in a very specific location and help carry out movement. But they do have these microfilaments, and the microfilaments can build and arrange themselves to create a barrier just inside that cell membrane and help support the structure of the cell overall and that nice round shape that we usually think of. The difference between the microfilaments and our skeleton has to stay in a fixed location. You can't take the bones of your arm and put them somewhere else in your body if need be. The microfilaments can actually break apart from each other and rearrange themselves somewhere else in the cell. So if you need an extra layer of protection somewhere, it can dismantle in one part of the cell and then reform in another part of the cell and then go back later on. So that microfilament structure, depending on what's going on, the cell can construct it where and how it needs it within the cell. Flagella and cilia. So these are projections on the outside of the cell. There is a big difference between, a couple differences between them. Number one, flagella, usually you just have one. There are a few exceptions, but generally if you have a flagella on the surface of the cell, there is just one, and it is primarily used for movement. Probably best example of where you would see this is the sperm cell. You have that tail that creates that whip-like action to propel the sperm. That would be a flagella. Cilia, on the other hand, are usually very short in structure, and you usually have a lot of them on the surface of the cell, and they are used for cleaning. So they wave back and forth. They can move the cell around, but their main goal is to make sure that debris doesn't build up around the outside of the cell. So you usually find ciliated cells in places like your respiratory tract and your nasal passages, where you're getting a lot of material that's coming in that you don't want in your body. And they just kind of wave back and forth, kind of like a broom, make sure that that sweeping action doesn't allow dirt and other material that you are bringing into your body to collect and build up around your cells. Golgi apparatus. Golgi apparatus, I already kind of touched on. It is another series of membranes that you guys have within the cell, but the Golgi apparatus is going to basically package the products that your cell has made, especially those products that are being delivered somewhere else. If you guys ever go on a trip, Generally, there is some preparation for that. Before you get in the car and leave, you're going to pack, you're going to get some things pulled together, bring snacks, things like that. You're going to get all of that ready to go. And likewise, that's what the Golgi apparatus does for the cell products. It packages and prepares them for the destination that they are going to go to. If they are going to go somewhere and they need energy along the way, the Golgi apparatus will pack on some carbohydrates to be used as a fuel source. So carbohydrates in the way of sugars, not carbohydrates as a marking flag. If it is going to be going to a location where it could become damaged, the Golgi apparatus will pack fat around it to help protect it. It might put on some protein. So if it's going somewhere and it needs to deliver protein, it'll pack it with protein and get it ready to go. So the Golgi apparatus is going to package and prepare the cell products that have been made for their final destination. Then we have the mitochondria. I don't know what it is about the mitochondria, but if people remember any of the organelles, they remember the mitochondria. It is the powerhouse of the cell. So last class we talked about cellular respiration, and I went through that process of glycolysis, Krebs cycle, the electron transport chain. That happens within the mitochondria. So we think about that ATP production, that fuel source, that is taking place within that mitochondria. Then we have the centrosomes. I'm going to wait on the centrosomes for when we get into our cell division talk. So the last one we have on there are the lysosomes. Lysosomes are the garbage cans of the cell. So your cells are constantly doing work, and you guys know that when things are being used on a regular basis, eventually they're going to wear down. If I buy a brand new pair of shoes, you wear them for about 6 months, things start happening. The shoelaces start to unravel, they get marks all over them, the treads aren't as good as they used to be. And that's the same thing with the parts of your cell. After they've been used for a certain period of time, they start to wear down, and some of them get damaged to the point that they're no longer usable. And you don't want that dead, damaged material to build up in the cell, so we have a waste system, and that's the lysosome. Now, the lysosome is filled with basically a digestive enzyme. And so if there's an organelle or a cell part that is damaged, it will get deposited into that lysosome. The lysosome coats it in a digestive enzyme, which eats away at it, very much like your stomach acid would when you eat food. And over a period of time, that dead, damaged material is broken down and removed from the cell. The next part talks about the different types of movement across the cell membrane. And so I told you guys earlier that we do have to move material across the cell membrane. Now, if you keep in mind that we are trillions of cells in size, this has to be something that we can do very efficiently, or we would not be able to get enough calories into our body to fuel that process. Trillions of cells that would need energy every time you wanted to move something in or out of the cell, there would be no way to keep up with that demand. So a lot of times when we are moving something across the cell membrane, it is using something called passive movement. Passive movement means that we can move it in or we can move it out, but there is no energy needed for that. So anytime you hear passive, that's telling you there is no energy requirement to move that material across the cell membrane. Now, there are multiple types of passive movement. You have diffusion. Diffusion is any type of molecule other than water. So if it's a sugar molecule, if it is an oxygen molecule, that would fall into the category of diffusion. The next one on the list is facilitated diffusion. Facilitated diffusion is going to move larger molecules. And so this would be where those protein channels would come into play. Those larger molecules that if we tried to push them in between the lipids would be so big that they would rip the lipids apart and break open the cell. Now we're going to use those protein channels. We're going to facilitate and assist with that movement across the cell membrane. And so that would also, still doesn't take energy because we're just moving it in between those protein channels to get it in or out of the cell. The next one that you have, still passive movement types, is osmosis. Osmosis is moving water. People love to use diffusion and osmosis in place of each other, and you can't. Osmosis is very specific. You are only moving water molecules. Diffusion is everything else. And then the last one is filtration. Filtration is when you have pores and you are using pressure to move things across those pores. We'll talk more about this. It comes up in our expiratory chapter when we talk about moving filtrate in and out of your kidneys. But those pores are just tiny little holes or openings that you have, and you apply pressure and you force that material either in or out of those pores, and that process is known as filtration. Now, you'll notice on these it talks about high to low concentration. And what that means is that whenever you have molecules of a certain type, in the case of passive diffusion, they always want to move from a place where there is a lot of them to a place where there is not a lot of them. If you've ever had a cup of milk and you've knocked it over, the milk does not stay in the cup. It spills out over the table, and it will continue to spread across the table until all of those molecules are spread out and equidistant from each other. And once they hit that point, the puddle is not going to spread anymore. But they went from where they were highly concentrated. They were packed into that cup. Once they were given the chance, they moved from high concentration in that cup to low concentration, spreading themselves out over the surface of that table. If you've ever sprayed air freshener tightly packed in that can, as soon as you spray it and release it, what do those molecules do? They float and they spread themselves out. They go from an area of high concentration to an area of low concentration. And that's what we see in passive movement. On the other side, you have active transport, and that is going to be where we have to use energy in order to move the molecules. And there are times in the body where we have to overload something to get a reaction to happen, to get a process to happen. Your nerve impulses have to have a buildup of certain molecules in order to get your nerves to fire. We have to stockpile that ahead of time, and forcing all of those ions into the same place, they resist against that, so it takes energy to pack them in and keep them there until your nerve needs to fire. Likewise, muscle contractions, same thing. You have to have a buildup of calcium ions to get your muscles to contract on call. And so before you ever need that muscle to contract, we have to make sure that those calcium ions are already in place and ready to go. Again, they don't want to be packed together, so it takes energy to move them into the muscle cell and hold them there until you're actually ready to use them. So in those cases, that would be active transport, because we are having to move these ions or move these molecules, pack them in together, and then take energy to hold them there until we're ready to use them. Endocytosis and exocytosis. Exo means exit, to leave. Cyto means cell. Endo means to move in. And again, we see cyto, which means cell. So exocytosis means something is exiting the cell. Endocytosis means that we are moving into the cell. The last two that you have on there, pinocytosis and phagocytosis. Pino means to drink, and then the prefix PHAGO means to eat. So in pinocytosis, you would be moving a liquid, and in phagocytosis, you would be moving a solid, something that would give the cell an energy supply. The last one that we're going to talk about, I'm not going to get into cell division for this conversation. That will come up later. The last thing I want to talk about is the slide that goes over hyper, hypo, and isotonic solutions. So we talked earlier about your cell being in a liquid environment. And because of that, you are going to have things floating around the outside of the cell. Could be oxygen, could be sugar, could be salt, could be a wide variety of things. The cell will always try and get the concentration inside the cell to be the same outside the cell. Now, I need to point out that that's concentration. That is not the number of molecules. And that's very, very important, because you could have a certain number of molecules floating around the outside of the cell, but it's the concentration of the liquid that is really what we're looking at. So in the case of hyper, hypo, and isotonic solutions, The cell is going to work to make sure that the concentration inside the cell is the same as the concentration outside of the cell. Now, in an isotonic solution, that means that the concentration inside and outside is the same. Iso means the same. The key feature there is that even though the concentrations are the same, you are still going to have things moving in the cell and moving out of the cell. It just happens at the same rate, and the concentration is balanced. And so you're not going to see any type of change happen within the cell. In the case of hypertonic, you think of a hyper little kid, their energy level is above or high. So if you look at the definition, it says a solution where the concentration of a molecule is higher on the outside of the cell compared to the inside. So concentration is higher on the outside. The cell has to do something in order to get that balanced. Now, it's not going to move molecules. It's not going to take those molecules that are making the concentrations off and move those. It's actually going to move water across the cell membrane. And so what will happen is it will take water from inside the cell and it will push it to the outside to try and dilute that higher concentration and get it to match the inside of the cell. So in a hypertonic solution, higher concentration on the outside, the cell is going to take its water content and flood the surroundings of the cell to try and dilute the outside and get the two sides of the cell to be in the same concentration. Think of a water balloon. If you have a water balloon and you start letting the water out of the inside of the water balloon, what happens to the size of your water balloon? It shrinks and gets smaller. And that's exactly what happens to your cell. Now, your cell will continue to give up that water content until the concentrations are balanced inside and outside the cell, or ultimately result in cell death. Hypotonic, on the other hand, now the concentration is lower on the outside of the cell. Hypo means below. So in this particular case, we are going to have water flood the inside of the cell and try and get the concentration inside and outside the cell to be the same by diluting the inside of the cell. Now the cell is taking on water, so it's like filling up that water balloon, and it will continue to take on water until the two concentrations are balanced, or if you've ever overfilled a water balloon, it eventually bursts and ruptures. And that would potentially happen to the cell. You could have the cell burst, again, resulting in cell death. So we'll talk a little bit more about hypo and hyper and isotonic solutions when we come back. There's a quick activity that we have with it as well to kind of give you a visual representation of what happens within those cells. So watch the video, take your notes, make sure that you bring those notes back with you at our next face-to-face meeting. Do not forget about our quiz over the anatomical terms. And then I will also post your assignment, so look in announcements for your assignment that you will need to bring with you to class.